We will not meet for class when we come today, on Friday. We won't be able to. I will have an online review, so you guys can jump into a Zoom and ask me questions there. Okay? Next week, we are going to start with chapter 6, but on Monday, I think I'll take a little bit of time to just kind of jog your memory on some questions and answer questions about what the exam looks like. Okay? So on Monday, we can have that discussion. There is an offset question. What does that mean? I don't know if you've ever heard of these, but I went ahead and it is open now. So this is a question that you can do outside of the exam time. There's no respondus on you. There's no one's looking at you. There is Turnitin, so I'll be looking for plagiarism. Mainly, I'm looking for you guys to write things in your own words. Okay. So let me open it. This is actually a question that is on the exam, but you get to do it away from the exam. So for this assignment, you'll describe the structural features of DNA that are responsible for these functions. Okay? We didn't sit through the process of DNA replication because that's from earlier stuff. That's a foundation that you should have, how is DNA replicated before coming into this class. But it's definitely something you can refresh your memory on. But we talked about things like proteins that bind DNA and features of DNA, like the shape and, things like that are responsible for binding. Okay? So I'm just going to leave that there. I don't want to talk too much about it. And then you guys can start looking at it now. So whatever if you look at your grade book, it's going to be in there as a 0. And whatever you earn for this is going to go onto your exam score. Okay, so that's what we mean by an offset. So it's going to offset this question. Okay, so if you go in to do the exam, if you do this assignment and you go in to do the exam and you don't answer that question, these points are going to get put into that spot for you. Okay? Does that make sense? So it takes a little bit of pressure off of you while you're taking the test. And if you're doing the offset question, I will say this. Go ahead and put references in there. Do some reading. Build a short essay that answers the question very thoroughly. Okay? Am I going to expect the same thoroughness on the exam? I might be a little more lenient on the exam because you're kind of just pulling from your own memory right then and there, right? Okay. Questions about that? I'm not even looking at you guys. All right. Yeah.
So in there you say that this is the same question, or like one of the questions that's in there actually be on our exam. If we choose to do the offset, do we have to do the question on the exam?
No, I would prefer that you didn't.
Okay.
Because then it would be a reminder to me to get those points over there. Cool. But if you do want to answer it because you're just like you can't leave a test with an unanswered question, it's okay. Yeah. Does that make sense? Yeah.
And I have another question. So for the writing assignment, it says one page, but it says double space. So now does it have to be 2 pages, or am I just reading into that? Sorry, it says it has to be 1 page, but it says it's double spaced. So do we have to have 2 pages for that? Does it say single space? I thought I read double. My bad. Just ignore me.
So you know what? I'm looking at you have a thorough description, discussion about gene therapy. But I tell this to everybody. I'm going to read through the content more than I'm looking at how long something is. Okay. So did you answer the question? Does it make sense? Did you say something totally off? That's really something that's going to look at. All right. And I'm glad you brought that up because there is something I wanted to share with that. So grading of that, there are 100 and some of you, and it's going to be sort of and I do want to read these, so it's going to be kind of a fast thing. I want to help you guys develop your writing skills. So what I will likely do is I will build a document that has common mistakes on it, common advice that I want to give to you guys, and then I'm going to end up posting that document and that's where you're going to get that feedback, and that's where you're going to get some pointers on how to improve writing. And you can go back and look at yours and think about what applies to you. And then your score is likely to be, you know, within a range depending on whether, using the rubric, depending on whether or not I saw, you know, 3 of these things of advice, these points of advice I had to give, or 1 or 0. Yeah, you had a question?
So did you say it opens on the test? Sorry, did you say it opens on Wednesday or Friday?
I can open it on Wednesday. It is scheduled right now to open on Friday while we are in class, but I would be fine to open it. But that's going to really, I would be fine doing that, but realize like you can't open it and then come to the review session. Because
Oh, I see.
when you open it, you have to complete it.
And then it's due Friday or
It's gonna be due Sunday.
Sunday?
Yes, so it posts, do I have the syllabus still up there? Do I say that on the syllabus? I never remember what I say. Oh, it just says that. Okay, and you probably can't see the instructions. So it posts Friday. You guys have until 11:59 p.m. Sunday to open it and complete it. So you get a couple of days. Thank you for asking that. Any other questions? Who feels really stressed out about it? Timeline though, let's just start, you got it on your radar, and meet with me if you have any questions, okay? Alright, let's go.
How many questions are there?
You know what, I can't tell you that, I actually do not know. I still have to make sure it's chapters, the right chapters.
So she didn't make it.
Okay? Thanks, those are out. Alright, let's finish up the information then for that exam. Let's go ahead and get into this. So, our last chapter for the exam, what is this, chapter 4? Is that what it says?
Chapter 5.
Is this really I think that's a typo. Huh. Am I off? Okay. Alright, so let's talk about genomics and proteomics. Am I off? Well, okay, I'm all thrown off now because I don't know if I'm off in the time. I can tell you that the quiz material covers what I'm talking about. It ends with what I'm talking about. I'll double check it, but I'm pretty sure. Okay. Whew. Be organized. Alright, so genomics and proteomics, we're gonna add on a little bit. We just went through those molecular methods. Now we're gonna kind of really look at we're gonna add a couple more and look at how we explore the genome and the proteome. So whole genome sequencing by next generation sequencing, that really changed molecular biology and the way that we start to answer questions and explore things. Excuse me. So we learned about next generation sequencing from some of our friends. Remember that we're gonna look at a couple of pictures of that. But that just really made things go a lot faster, and it just streamlined the process a little bit. So there was no more one gene, one graduate student approach. So I don't know if you've ever been in a research lab, but you know, some research labs have a nice strategy where they're like, you know what, you get to study this protein, and that's your job. And so that's what it was to understand a single gene for a time, a single gene. Now, does anybody remember how many genes are in the human genome? Billions. Yeah. 3 billion genes are in the genome. So just think about that. Like, how many graduate students would it take for us to figure it out, right? A lot. Okay, so the new genomic approach gave us a lot of data, you know, from a single graduate student. So it really made things possible. It made the end goal possible. This is a lot of information, so much information. So analysis really, you know, this was not made possible without bioinformatics. So that computational approach to handle these long sets of data. You can't do this by hand. You definitely can't. So genomics and these omics are really important, or really their goal is to quantitatively understand the system. So we're asking, you know, what, we know, we don't just go from, we know the sequence, we go to what's being expressed and why. Okay, and that's on the multiple levels of gene expression. Remember what a reductionist approach is? It's where you tease out a single thing and you study that single thing. So the Human Genome Project was complete in 2004. It's been a little while, but this is a timeline of how these things happen, and it's just an interesting timeline because when you look at this, you can start thinking about, just now that you know these methods, you can think about what it took for somebody to sit there and figure this out, right? Or for people to do that. So it was in the 1970s, 1977, that the first viral genome was sequenced. So why is this the first one? Because viral genomes are really short, right? Just like 1,000 nucleotides or something like that. Okay. This was really cool. We figured out we could sequence something like that. But then, of course, in science and medicine, we're a little like, we're really, into ourselves. But not that. But we really are trying to solve problems, the human problems, right? So the Human Genome, HGP is the Human Genome Project that was launched, and it was estimated even at that time to take about 15 years. So in 1995, we had our first bacterial genome sequence. This is, 4 million genes maybe that we needed. So we were getting bigger, capable of more. 1997 was yeast. Now we've got a eukaryote that we see the genes for eukaryotes, and we can start understanding some genetic information differences between the prokaryote and the eukaryote. All right, it was in 1999 that the big accomplishment was chromosome number 22. So human chromosome number 22 was sequenced. How many chromosomes do we have? Does anybody? I never remember this. There
46.
are 23 pairs of chromosomes, so 46. So we got one in 1999. Okay, so then in 2000, a more complex organism, Drosophila melanogaster, the fruit fly, was sequenced. And 2001, we had the draft of the human genome finally published. A draft. There were mistakes. And why would it be a draft? Because if you've ever gotten the sequence back, sent something out for sequencing and gotten it back, the reality is you get letters and X's and gaps and things like that. It's not perfect. There are imperfections in it. So take some repeats over this section to really figure out what it is. So the first draft was in 2001. So 2002, we had mouse and Plasmodium genomes sequenced, so a little higher order. And then it was 2003 that finally the human genome was finished. You guys have an old slide. I actually updated this morning. At this point, now we have like 27,989 eukaryotes in the database, and we have 513,032 prokaryotes in the database. If you ever want to go explore that, this link actually, it does work. It takes you to the NCBI genome database, and you can click on a genome and you can see you can scroll through all the letters and see the sequence of stuff. There's also pretty nice graphics sometimes that kind of organize things too. So what are we talking about? Let's really think about what happened. So at first it was really hard. So like every year, I should make a point, the information basically doubled in 2 years. So like this stuff, like right now we are, like in the last slide that you had in 2019, and there was like 9,000 eukaryotes or something like that on there, and now it's been, just a couple of years, and now we have 500,000. It's like, it's really going very, very fast now. It wasn't that fast in the beginning. So we had, so this is the way it was done. We had those bacterial artificial chromosomes vectors. You remember the vectors we introduced a few talks ago. And so these are, bacterial chromosomes that are, were manipulated so that we could insert our genes of interest in that. And what we did is we made these libraries. So if you wanted to sequence E. coli's genome, you took that genome, cut it up into pieces, and then you ligated those pieces into individual vectors. Okay. Right? So why not the whole thing? Why not the whole thing? The reason we don't just take the E. coli chromosome at this point, we didn't just take the E. coli chromosome and run a PCR reaction for sequencing, because one, we didn't know where to put the primers. You guys remember how the PCR works? You gotta have a primer, so we didn't know what primers or where to put them, right? We don't have that information. The other reason is because PCR, at this point, even now, if you run a PCR reaction, your sequence is probably good for about 1,000 nucleotides. After 1,000 nucleotides, our machinery starts to fall off. Our DNA polymerase, it peters out, basically. It doesn't do well. And that's just in vitro. That's just it's an artifact of being out of the system. Okay? So, alright, so these individual pieces, so you take the genomic DNA, you cut it up, and then you put each of these little pieces into its own vector. So now the BAC, we know where we can put primers. We know where the ends are because we use the BAC's sequencing. And we put those, the forward and the reverse, in there, and then we see what we get when we amplify. Alright, so then we get the sequences, we cut them kind of in these, many copies of them, we cut them in these staggered ways, or we use the primers from the BACs, and we started to line, we get all of our little fragments and we sequence them and get the sequences on the computer, and then we use the computer to show us, find overlaps, and then we would build a contiguous sequence from those overlaps. And then eventually we got the whole E. coli chromosome. Okay, once we stitched them all together. So that's what we mean. So we fragment the BAC sequences in small overlapping reads. That's what these little overlaps are. Then we assemble the reads into contigs, and then we map it. We are able to map it together. You guys will actually, you guys, if you took Biology 1, you did something like this in one of your labs. You took your sequences and found the overlaps, and then you wrote the whole sequence. Alright, so here's the stack up of the different genome sizes. Here is that, what was the first one? We had a viral genome. Here it is, 1.8 million bases. 1.8 million bases, so this is each individual nucleotide. And then E. coli is about 4.6. And then look at humans. So many of them. Okay, so then also included in this table is showing us the number of protein coding genes. So this is how long the sequences are, the total set of genome nucleotides. But then this is about how many are actually protein coding genes. So this is how many genes there are. Each gene's like thousands of nucleotides long. This is how many nucleotides there are, and then this is the number of genes within that length. Okay, so 90% of the viral genome is actually coding for protein. 88% of the E. coli genome is coding for protein. Look at us. 1.2% of our genome actually codes for protein. As of this time, our understanding at that time. It's crazy. It's really crazy. So another point is the number of genes is not necessarily equal to the complexity of the organism. Okay, so the big surprise, right, was that there were only 21,000 genes in the 3 billion nucleotides of the human genome. and only 1% of the genome is coding. Okay? So humans have 21,000 genes, but apples have 57,000 genes. You know, which one's sentient? Which one's really aware of itself and, you know, all of that stuff. So it's not necessarily, you know, the number of genes does not equal the complexity of the stuff. Okay, so this doesn't mean that we are less than a virus or something like that. Okay, why does a virus have so much genome that is protein coding? Because it doesn't even have the full set of genes to exist, right? So it's got the genes that it has are specifically for, like, these are the key proteins that are needed for this thing to hijack its host. Okay. We will get into the human genome a little bit more and talk about the, you know, what are all the pieces, but we have so much, something that just to kind of seed into your mind now. Something that is really kind of cool about the way that we do it, the way humans do it, is our gene expression is very regulated in time, and some of that has to do with a lot of this, you know, the other percentage that isn't included in the genes that are actually coding for the protein. Okay, so you've got your open reading frame that codes for your protein, but you've got all this other stuff outside of that that is regulating when that's on and when that's off. Yeah, so that's a big deal too. And we have a bunch of copies of things too that have evolved with us. Okay, so about half of the human genes are common to all vertebrates. Typically, this is just a little cool Venn diagram showing you which, you know, the number of genes that we share with chickens and zebrafish, mice, and all of that stuff. I can tell you something really interesting. Well, let me make this point, and then I'll give you the interesting thing. So typically, these genes that you're seeing that are shared, these are typically genes that are involved in building or coding for the proteins in metabolism. You know, all of those enzymes that catalyze those reactions and all that stuff. Also, the gene expression genes are pretty conserved among all the different species. So the genes that code for DNA polymerase, RNA polymerase, the genes that code for transcription factors, things like that. The helpers, the machines for actually expressing genes. So I'll tell you an interesting thing from research times for me. We studied, you know, the human gap junction problem in cancer cells in my PhD lab, and we actually, when we introduced, like, we wanted to increase the expression of our protein connexin 43, we would actually introduce a chicken gene sequence, and it would do everything that we needed it to do. So there is conservation of gene sequence among different things, but we sometimes we chose the chicken because it was like we could follow that gene. You know, there was a difference, slight difference between the two. Okay, so genomics has allowed us to compare the genomes and get some information. So modern humans and chimpanzees are about 99% identical. Humans and Neanderthals are 99% identical. So you can get these comparisons. Okay, it's really interesting. There's clear physical differences. So here's the tree, the clades. It's really interesting because there's like clear physical phenotypic differences between those organisms, right? And that's made possible by, you know, there's only 90 genes that have differences in coding between Neanderthals and us, right? So 90 genes have been changed, and then there's a clear difference, or there are 90. Shouldn't say have changed. There are 90 genes that are different between the two, and there's a clear difference in the whole developmental makeup of those two things. So it's kind of a neat thing that you can do when you can see the whole genome. All right, so next generation sequencing, we're really excited about that. It did make a really big difference in allowing us to see. So remember the backs. You know, some graduate student had to get a genome, cut it up, put it, each one had to be cloned into a vector. That is a feat in itself. And then sequenced, and then the computational stuff where you had to sit there and look at these sequences and put them together, and all of that. And by the way, with cloning, like you gotta cut DNA, clean it up, run it on a gel, clean that out of the gel, put these two things together, ligate them, grow them in another organism to make a bunch of copies, all that stuff has to be done. Next generation sequencing was really neat because this eliminated the gel electrophoresis step in a lot of the steps. So just a reminder what we're talking about, we take the cell DNA and we fragment it, so cut it up with restriction endonucleases, and then we add linkers to the ends to provide basically an attachment site for the chip, and also by adding our linker, by adding our piece, now we have a piece that we can develop a primer to. Does that make sense? So now we have a known primer that we can use. Alright, so these are all of these fragments are attached to this little glass chip, and then we run the amplification, the PCR reaction on these things attached to the chip. So each fragment is amplified by PCR, and we make a cluster. We make a cluster of that fragment right there on the chip. Why do we care about the cluster? So remember how sequencing worked? We had the modified dideoxy that terminated. So right there in that cluster, the way that it's run is you have those different fragments in the cluster. If you think if you were to blow up the chip and look at that. And you can have your color labeled nucleotides, and then right there in the cluster, because you have many repeats of that single piece, then each one can be read with confidence, right? Because we have so many little copies there on the glass chip. Alright, so with the sequences, you run sequencing on each cluster, collect the dideoxy termination data, and you have a huge number of fragment reads all on this little glass chip, right? So now instead of taking every gene, putting it into a back vector, doing all that work, it's just on the chip. I shouldn't say it's just on the chip. It's still hard work. It's still something you have to do, but it is a lot faster. It's much niftier. We still have to assemble contigs because we did fragment the genome, but that's the easy part. That's the easy part thanks to bioinformatics. Thanks to the ability to look at this using a computational approach. Okay, so just some more history. So the first individual human genome to be sequenced was Craig Venter. He was the entrepreneur that really drove a lot of the companies that developed these tools and the methods and all of that stuff. And James Watson. Is that Watson Crick, really important DNA person. So that was, it really took a lot of money, a whole lot of money to do. And then, as of 2015, 2016, the cost to, now that the tools have been developed, the companies exist to make the tools, it was about $1,000 to sequence a genome. I tried to look up this morning what it costs now. I couldn't, you have to join have an account and all this stuff, so I didn't figure it out. But I imagine it's not as expensive. So now, what's cool about this, the major breakthrough, the major importance or significance of this is now that you can get your individual genome sequence, now you can really start to think about personalized medicine. Okay, what is your mutation that causes you to have this thing? Okay, so just an example, personal one for me. My father-in-law is the healthiest person that I know, but has had 4 heart attacks. He happens to have a very small mutation in a gene that codes for an enzyme for lipid metabolism. And, so knowing the Thank You Human Genome Project, we know what those genes are, and that multiple people have had this and that this is what has happened, right? So now we can link all this stuff together. Okay, so, let's see, so now we're gonna look at look at some methods. So we got some studies of gene expression. So again, next generation sequencing really revolutionized the study of gene expression. So let's look at we're gonna look at a couple of the traditional ways that the whole genome was looked at. So we looked at the whole cell gene expression using microarrays. So I highlighted gene expression for myself to remind myself to say this. When we're talking about gene expression now, we're flipping. We're flipping from looking at what are the nucleotides in the genome. Now we're gonna talk about what is being expressed and when. So RNA. So RNA and protein. Okay? So we have microarray, and then now we use gene expression looking at RNA-seq. Okay, so this has allowed us to have a new area of study, which is transcriptomics. What is the RNA that's coming out of this tissue cell, and why, when? Proteomics, what are the proteins that are coming, right? Okay, so transcriptome is all the RNAs that are transcribed in a cell. I changed this slide this morning so you don't have this picture, but what this is right here is a heat map of the RNA that is being expressed in different samples or different cells. Okay? So the microarray is this cool technique or this cool approach where you have oligonucleotides printed onto a glass or silicone chip. So we know the genome now, right? So now we can print the DNA gene sequences on a chip. Okay? So kind of just jump back here again, look at this picture that. Okay, instead of getting them from a cell, we actually synthesize them on the chip. And they're floating like that off of that. Okay. Then let's see, so every spot on your glass chip is going to have a single oligonucleotide. So what you can do is you can use these microarrays to compare gene expression between two cell types. Something that you got to picture before even thinking about that is, so what we can do is you've got these on this chip, you can take your cell, bust it open, harvest RNA, and then you can ask the RNA to anneal to what's on the chip. Okay, so you basically flood the chip with the RNA. And why would they anneal? This is really important for our sex questions, right? Because they will hydrogen bond with the complementary nucleotide bases, right? Okay, so now imagine you can take one cell type and a cancer cell and a non-cancer cell. You can use the same chip and compare what genes are being expressed between those two different scenarios. You can have a drug treated and a non-drug treated, and you can compare the genes that are being expressed between those two things using the same chip, right? We do sometimes need reverse transcription to generate cDNAs. RNAs are not really stable. I think I mentioned that before. We are dripping with enzyme to chew up RNA. Sometimes it involves isolating the RNA and then reverse transcribing it, getting rid of those U's, putting the T's back in, and the dideoxy rather than the ribose sugar. And then that's a stable molecule we can hybridize to our chip. So that's what you're seeing here is, let's see, so the greens, we didn't have gene expression of those genes. The reds, we had a lot of RNA copies. They annealed to their sequences. So here's another look at that. Here's a cancer cell isolating the mRNA, and the normal cell isolated the mRNA. We converted the mRNA to cDNA, and then on this chip we have every little dot is a known gene sequence. So then we pipette the clear liquid onto the chip, let it see what binds. And usually by the way, we do these cDNA probes, we do the reverse transcription, and in that process of reverse transcription, we can add on a fluorescent gene too. So now you can see, you make a copy of your RNA and it also is tagged with a fluorescent molecule. So then you get to see, is there some red or is there no red fluorescence? Okay. If there is red fluorescence, then we got gene expression. We got RNA of that. Does that make sense? This is one of those hard things to kind of picture. Okay. All right, so RNA-seq is something that helps you to directly sequence DNAs, and this is where next-gen sequencing comes in again. So this takes, it reveals sequences of all mRNAs in a cell. So your cellular mRNAs are all reverse transcribed to cDNAs, and then you can analyze them using next-gen sequencing. So we take reverse transcriptase, we harvest the RNA, reverse transcribe, and then we put those onto the next-gen sequencing chip. We amplify, read, and then we know all the sequences. Okay. Oh, I should have went to the picture. So here's our mRNAs. We reverse transcribed into cDNAs, and on the chip we can then start to, using the intensity of the reporter, we can see how much, how many copies did we get of the P53 RNA. How many copies did we get of the connexin 43 RNA? All right, proteomics. Proteomics is where we go, we start to go to the protein level. Remember, there's only a certain, this is a big feat, and truthfully, it is still, if you're trying to understand the whole genome and all that, remember it's still just a really small drop in the bucket. Like 1.1% of the genome gives us proteins, or something like that, right? But anyways, this is still a large-scale analysis, because we're gonna look at all of the proteins that are expressed in a certain tissue or in a certain scenario condition. So the goal is to identify all proteins expressed in a given cell, and that is the proteome. Proteomics is the analysis of it. Proteome is the set of genes expressed in proteins. So the number of proteins expressed in a cell is greater than the number of genes transcribed. This is a really interesting thing. Why would that be? So we only have a small set of genes that give us proteins, but we actually get more proteins than even that number. That's really interesting. Here's the reason why, because there's a couple of reasons here. So some genes can be expressed to give you a distinct mRNA, but they can be alternatively spliced. We get different polypeptides because those mRNAs are alternatively spliced. So remember the big difference between eukaryotes and prokaryote RNA expression is eukaryotes have introns and exons in them. And those introns and exons can be, for maybe in the heart tissue of this gene, we take intron, we are reorganizing introns 1. I always say the wrong thing, but we're reorganizing, P's 1, 2, and 5. And that's going to be in this tissue. In this tissue, we're going to reorganize 1, 2, and 6. Okay. So that's what alternative splicing is. So from a single gene, you can get, 4 or 5 different mRNAs that are going to give 4 or 5 different proteins. Okay. Your proteins also can be modified in various ways. So we can add those functional groups, phosphates, methyl groups. We can add, fatty acid chains, things like that. So then we essentially get different proteins with different functions because they have these different chemical properties. All right, one of the first techniques to separate our proteins and start to look at all the proteins in a sample was the 2-dimensional gel electrophoresis assay. And this is one where the proteins are first separated by charge and then separated by size. Why did we have to go with this? Because proteins all of our proteins have different shapes to them. They're going to have different densities to them. So we can't just do a single measurement, right? It's not as easy. this is what it looked like. Imagine figuring this out. What is all this? Okay, there's a couple of things you can do. You can look at these dots and you can start to ask yourself how many proteins are in this sample. Like if you have an identical a tissue culture of identical cells, then you know all of these pancreatic cancer cells, these are the proteins that are in them. Okay, you could make a comparison, like treat them with a drug and then see if some of these dots go away, or see if you get new dots. So it looks like a mess, but there's a lot there that you can still figure out. Right? All right, now a better way is mass spec. And mass spectrometry is where you take a protease, you take a protein that's cut up into small peptides. They're ionized, so they are given a charge. They in the process, and then they're sent through this chamber that reads things like their, what is their charge, what is their size, things like that. And you get what's called the mass to charge ratio of each peptide. And it's a signature that can be used to figure out how many proteins are there. And then also get a little more detail of what the proteins are, what functional groups they hold or something like that. Okay, so the mass spectrum is, we can compare it to a database of known spectra, with further deeper dives into, we take first mass spec, we figured out these are the proteins there, and then we started to do functional assays and figure out what they were doing. Okay, so here is traditional mass spec, where you have your purified protein. So this is on this side, we had to have purified protein, broke it up into pieces, ran it through that chamber, and we got this spectrum of mass to charge ratio. And then you literally compared this little graph of peaks with other known things to figure out if you had, do you have a tyrosine? Do you have what are the amino acids that you're looking at? Okay. This one over here is shotgun mass spec. It eliminates the need for purification. You just basically put all your peptides in, get your mass to charge ratio. There's a collision-induced fragmentation and your spectrum of mass to charge ratio. And the spectrum allows you to build the amino acid sequence. So here's our proteins. So protein localization is one really nice thing that we have to figure out. Okay, so the number of proteins identified in the plasma membrane are 860 to 2,000. The Golgi has about 400. So this is reductionist. Remember our cell fractionation? You can do this type of stuff on the Golgi only, on the mitochondria only. Okay? But we also want to know, not only do we want to know where they're located, we want to know what they're interacting with. Okay, do I have 5 minutes left? Is that clock right? Okay, all right, because I'm going to get to our, what's on the quiz. So the protein interactome is basically the assays where we're looking at, what proteins are connecting with what proteins, and then we start to answer questions like, if they're coming together, what is the outcome? Like, what is happening? So we have a systemic analysis of complexes and their interactions, and that is the overall goal of proteomics. So one thing that we can do is immunoprecipitation to isolate proteins and protein complexes. This is also called co-precipitation. All right, the way that this works is you can take a mixture of proteins. You can incubate, and you can take a bead with a known antibody to a known protein. So say you want to look at the EGF receptor, the epidermal growth factor receptor. You can have an antibody to the EGF receptor hybridized or chemically attached to a bead, to some surface. You mix these beads with your mixture of proteins and you capture those proteins. So you will capture, say you're looking at the EGF receptor, you've captured all of them. Then you can take your antigen and antibody complexes and you can release all of the proteins and you can use mass spec to see, did I get the EGF receptor, and then did I get something else? Did I get another protein attached? So your immunoprecipitate, if I were to redraw this, I would show these green proteins immunoprecipitating with the the antibody, right? But then I'd also add on a binding partner. And that's what you can look at. That's what you can figure out. The binding partner is there. All right, here, yeast 2 hybrid. Yes, there's a question on your quiz about the yeast 2 hybrid assay. So, the yeast 2 hybrid assay, this is a method to show, very cool method to show protein-protein interaction by really pulling it out of the cell and putting it into a system that is not natural to it. So the way that this works is you have two different, you'll take your two different you'll take your cDNAs and they're fused to two distinct yeast genes. Alright, so you have one yeast gene is going to code for a DNA binding domain, and the other is going to code for a transcription activation domain. So these domains are the zinc fingers, these things that are going to bind the major and minor grooves of the DNA. These are the actual DNA binders, the proteins that bind the genes. They're going to be reliant on those grooves and the nucleotides that they recognize, they bond with. Okay, so the two human proteins are fused to these two different proteins that are for binding the DNA. And then if they interact, only if these two proteins interact, will the DNA binder and the transcription activator, only then will these two things come together. And then you'll get gene expression of the reporter gene. Okay, so here's a picture of it really quick. Whoop. So here it is, so you've got your yeast domain 1, the DNA binder, and your yeast domain 2, the transcription activator. Here is our protein A that we think is interacting with protein B. We put them into the yeast cell system. We have some gene that will only be expressed by this binder and this activator. Is reliant on this specific DNA binding protein and DNA and transcription activating protein. Alright, so notice this one does not actually bind the DNA without its friend. So only upon binding of the two questionable proteins will you have binding at the promoter site and then activation of gene expression of your fluorescent gene or something like that. Does that make sense? I went through it kind of quick. We'll bring it back on Monday so that you're really comfortable with it.